Before going to our speaker's presentation, I would like to welcome Chris Graney to have give us a keynote talk. So welcome, Chris Graney.
Thank you. Yeah, thanks for the introduction. And I guess I should stop. Yep. You can see that all okay?
Yeah.
Fantastic. So thank you so much for that lovely introduction. It's a real pleasure to be here. I'm joining today actually from San Diego, having just come from Australia yesterday. And what I'm going to be talking about today is this evidence that Antarctica is a little bit different from other continents in the world in that we believe that it's principally, not entirely, but principally driven by chemical energy sources, especially atmospheric trace gases like hydrogen, carbon monoxide, and methane, rather than photosynthesis like in other continents of the world. And the first thing I just wanted to talk a little bit about is why this is. And I'm going to focus initially on our findings in the Mackay Glacier region with a lovely photo here from one of our collaborators, Ian Hogg. So when you look at Antarctic ecosystems, typically in the ice-free soils, we've seen this, but also others like Craig have seen this. There's often hundreds of bacterial species and millions of cells that inhabit every single gram of these Antarctic ice-free soils. Yet when you look at the capacity for photosynthesis, so key taxa like cyanobacteria as well as photosynthetic eukaryotes, often they're incredibly low abundance, at least in the exposed soils. They're sometimes present in the hyperliths, indicating there's a low capacity for photosynthesis. And consistently, when we do activity measurements to look at photosynthesis, there's basically no activity going on in a majority of soils, at least in their native dry states. Yet nonetheless, there is life there, so indicating there is some capacity for other energy generation processes occurring. So in Antarctic soils, there's multiple stresses that life has to deal with. They have to deal with the freezing temperatures. They have to deal with polar darkness, which is limiting for photosynthesis as well, often high salinity, and possibly high UV radiation is a significant pressure as well. And so the key question is, if photosynthesis isn't possible, how are these microorganisms in this poly-extreme environment actually still surviving, if not thriving, in this continent? And so our big question was, is chemosynthesis the missing link? So when we think about microorganisms' nutritional strategies, they're actually incredibly diverse, both within and between taxa. Many can photosynthesis, just as plants do. Others are capable of using organic carbon, such as animals, just like animals, sorry. But many are also capable of using inorganic compounds, whether that be hydrogen, carbon monoxide, nitrogen compounds like ammonium, sulfur compounds like sulfide, metals like iron as well. And yet, when we think of chemosynthesis, we normally think of geothermal systems like these beautiful sites, such as in Yellowstone or Rotorua and whatnot. And we don't normally think of Antarctica as a continent where there would be much chemosynthesis, with the exception of certain geothermal regions. And this is because obviously when there isn't this geological activity, it's not going to be these key energy sources like hydrogen or sulfide, often high abundance. So nonetheless, we believe that chemosynthesis is very, very significant because of the fact that there are still actually plenty of chemicals, but actually of atmospheric origin instead of geological origin. So the first bit of evidence that we got for this was this collaboration that was published in Nature with Belinda Ferrari and Phil Hugenholtz, and then we did a follow-up study in PNAS with Don Cowan. And there we took, in both studies, a genome-resolved metagenomic approach to getting a first insights into what are Antarctic soil communities actually capable of doing. And we were looking at a range of metabolic capabilities spanning use of organic compounds, light and inorganic compounds. What we can see is that when we look at most of these metagenomes, in this case from the Mackay Glacier regions, we see that generally the capacity for photosynthesis is low. There's some capacity for light capture using rhodopsins, but actually oxygenic photosynthesis is very, very low. But nonetheless, there was this capacity to use inorganic compounds like sulfide, ammonia, and iron, which are probably found, which we know are found in the rocks of these ice-free soils, the lithic substrates. But then there's also capacity for using trace gases as well. We saw that for some of the sites, there were extremely abundant methanotrophs that are capable of using atmospheric methane. But by far the most abundant metabolic capabilities other than organic carbon usage was associated with using these two trace gases, carbon monoxide and hydrogen. So we saw that the carbon monoxide dehydrogenase was found typically in about 60% of the community. And this carbon monoxide dehydrogenase oxidizes CO to CO2 with the electrons being transferred into the respiratory chain. We also observed that the capacity for hydrogen oxidation via this group 1H and group 1L nickel-ion hydrogenase was highly enriched too, with about up to 100% of the community being capable of using hydrogen as an energy source, with hydrogen being oxidized by these hydrogenases to release electrons and protons, with the electrons being used again for aerobic respiration. But the big conundrum is where they're getting the hydrogen, carbon monoxide, and methane from. And it actually turns out they're getting it directly from the atmosphere. So when we think of the atmosphere, we normally think of it as a source of oxygen for aerobic respiration, nitrogen for nitrogen fixation, and carbon dioxide for carbon fixation. But there's actually a bunch of trace gases also found in the atmosphere at fairly low concentrations. So usually in the sort of part per million range, including methane, hydrogen, and carbon monoxide. And these have three things in common. They're ubiquitous, they're always going to be available at least in atmospheric concentrations. They're high energy, so by oxidizing them, it's possible to yield quite a bit of free energy and ATP for the cell, and they're also freely diffusible into cells. But the reason that hydrogen and carbon monoxide are particularly favored compared to methane is they also have low activation energies, meaning that you don't need to invest oxygen and reductant, as is in the case of methane, in using them. So they turn out to be incredibly highly dependable energy sources for life in these energy-starved environments. And so when we do activity measurements, we can confirm that in Antarctic ice-free soils, the levels of hydrogen and carbon monoxide oxidation we see is pretty vast. So this was work done in collaboration with Carlo and Matt, and the three of us basically came up with a bet of how rapidly would these soils consume hydrogen and carbon monoxide. So we wanted to test if this genomic capacity could be validated. And because the Antarctic soils, we thought it's going to be slow. I think Carlo said 3 months at best. I had a slightly more liberal prediction, but it turned out that for hydrogen at least, within about a day, nearly all of the hydrogen that we added to these soils was gone. So in this case, we had a serum vial, 120 mils, with 1 gram of ice-free soil from, in this case, the Robinson Ridge and Adams Flat regions of Antarctica. And then we added quite a bit of hydrogen, 100 ppm, in this case, and measured over time by gas chromatography how rapidly this hydrogen was gone. And you can see within about 24 hours, it all gone. Carbon monoxide oxidation was a bit slower, but still very, very significant rates. So keep in mind that well into the 1970s, ecologists were saying Antarctica is essentially a defunct continent, at least some ecologists. Then through the work principally led by Cary, and Don Cowan, these molecular surveys revealed actually there's plenty of life there. But then when you look at this really important biogeochemical activity that people previously overlooked, it turns out that this life isn't just there, but it's incredibly metabolically active as well. And through our work with Sean, we're also able to show that carbon dioxide fixation also occurs in these soils. And instead of being driven by photosynthesis, so by light, it's actually primarily driven by H2 gas in these Antarctic soils, which is, again, quite a paradigm shift in how we view life in this continent. So why do they use trace gases? Well, it actually yields 3 different things in short supply in the continent. So first of all, energy. So we can actually do thermodynamic modeling based on the rates of hydrogen oxidation and carbon monoxide oxidation we see versus the proportion of Antarctic microbes in soils that can use these gases. And based on this, we predict that about essentially all of the bacteria that can use these gases are capable of, at the very least, meeting their maintenance energy needs in these gases. But we think that the ranges in some soils are sufficient to also sustain growth, slow growth, but growth. We believe that this process is also incredibly important for supplying carbon in the continent. So these Antarctic microbes, as I just shown you, use electrons from hydrogen and possibly carbon monoxide to fix carbon dioxide into biomass via the carbon-Benson cycle. And to do this, they use a slightly different variant of rubisco to what plants do. They use the type 1e and sometimes type 1a rubisco instead. And the final thing, which eluded us to until recently, is the fact that this process is also almost certainly a quantitatively significant source of water in this desiccating environment. So when you react hydrogen with oxygen gas, the end product is water. We know this from hydrogen-powered cars. Cells do exactly the same thing. They're producing cytosolic water in this process. And based on back-of-the-envelope calculations, a standard E. coli cell, if you assume that every single bacterium in Antarctica is the same size as E. coli, which of course they're mostly going to be much smaller, then through this process alone, these cells can completely rehydrate within about 15 days. So we think this is really important for avoiding desiccation. So taking a more detailed view, this is based on genome-resolved metagenomics. And here we can basically reconstruct the dominant energy generation and carbon fixation pathways in this continent. And we see that when most bacteria will have the capacity to use organic carbon, of course, this is in short supply. So they have to use these trace gases, hydrogen and carbon monoxide, as energy sources instead. They freely diffuse into cells. They're oxidized by these high affinity hydrogenases and carbon monoxide dehydrogenases. This powers aerobic respiration, yielding water, but also allowing the generation of a proton-motor force that powers ATP synthesis. And a subset of these bacteria will also fix CO2 into biomass. We don't know exactly how to do this, but we assume that this reverse electron flow that allows them to generate reductant for this process. And through this activity of this type 1e rubisco, this allows these cells to at the very least maintain their carbon levels, possibly generate that carbon for growth as well. With, in this case, the primary producers primarily being actinobacteria, which we don't normally think of as primary producers, with these two other bacterial phyla, Dormibacterota and Aremiobacterota, also really important, with Belinda discovering these two phyla for the first time in this Nature paper. So one of the things that when we released this, when we published this Nature paper, I think wasn't highlighted was the only reason that we realized Antarctic bacteria could live on there was only from having the precedent previously of pure culture studies. So we didn't just come out of this from nowhere, but actually had a bunch of pure culture studies in non-Antarctic bacteria, which showed that the atmosphere was actually a relevant energy source for microorganisms. So during my PhD with Greg Cuck, I was able to co-discover that Mycobacterium spermatus is capable of using atmospheric hydrogen and later carbon monoxide as alternative energy sources. And this made these discoveries in Antarctica possible. So just as a bit of evidence for this and explaining again why bacteria use trace gases, just wanted to briefly cover some of the background here. So this is showing our work on carbon monoxide oxidation, very similar work we've also shown for hydrogen oxidation. This is led by Cary and Paul. And what we can see here is that Mycobacterium spermatus oxidizes carbon monoxide to subatmospheric levels using carbon monoxide dehydrogenase. If you knock out the gene for this process, it no longer happens. This process is productive, so carbon monoxide stimulates aerobic respiration, doesn't inhibit it like it does for us. And this process enhances long-term survival, so it's upregulated during survival. And if you delete the genes responsible, it results in about a 50% reduction in long-term survival. We see very, very similar things for hydrogen oxidation instead, meaning that when bacteria in a carbon-starved environment, these trace gases become incredibly important. We believe this is a universal metabolic strategy. So when we do genome surveys of the carbon monoxide dehydrogenase as well as the hydrogenases, we see they're incredibly widespread across soil, marine, host-associated bacteria. And we've been able to experimentally show all of the following isolates are capable of using atmospheric hydrogen. And these span a whole myriad of lifestyles. They span everything from geothermal thermophiles, such as Thermomicrobium rosium or Perinomonas methylalpha-genus, right the way through to psychophilic Antarctic isolates like Heminobacter roseus-salivarius. So here you can see for this Antarctic isolate, this bacterium will consume hydrogen to subatmospheric levels using, in this case, a group 1L nicoline hydrogenase. And we also see for a range of different lifestyles too, spanning methane oxidizers, nitrate oxidizers, and some iron and sulfide oxidizers, as well as more conventional organoheterotrophs like Mycobacterium. And we've also looked in great detail at the biochemistry of this process, and this shows our unpublished structures of the hydrogenase and carbon monoxide dehydrogenase that are driving these processes. This isn't a biochemistry talk, so I won't go into any detail about this now. Just several remarkable features about these enzymes that allow them to function in air, where they're able to consume tiny concentrations of these substrates. They actually, in their purified state, are extracting energy from atmospheric hydrogen and atmospheric carbon monoxide, but also tolerate O2 gas, which at least in the case of hydrogenases tends to be a poison. And this is beautiful work led by David, Ash, and Reese that I hope will be out later this year. So the final thing I wanted to talk about was how does this stack up globally? And I'm going to first of all talk about other soils, and then I'm going to move on to other ecosystem types. And so the first thing I wanted to talk about was this global survey that's been led by an exceptional PhD student in my team, Bob Lung. And here he has, in collaboration with numerous people across the world, compared what is the microbial biodiversity and metabolic capabilities of bacteria in polar deserts, including Antarctica, various hot and cold deserts as well. And then he's compared that systematically to the communities inhabiting agricultural, forest, grassland, and wetland ecosystems as well. And when you take this sort of more global perspective, what we see is that in general, in terms of metabolic capabilities, deserts tend to have quite similar capabilities, whether they're polar, cold, or hot deserts, with, again, a limited capability for oxygenic photosynthesis, but a very high capacity for using these trace gases. But interestingly, the levels of trace gas oxidation vary depending on the extent of aridity in these deserts. So we showed this previously in published work in the ISME journal, going from an aridity gradient in the Negev desert region. But here we also see that the soils with the highest capacity for atmospheric chemosynthesis, so using the atmosphere as an energy source for carbon fixation, where in fact was in fact the Atacama Desert and some parts of the Gobi Desert, indicating that in these really harsh environments too, this atmospheric chemosynthesis is incredibly important, but very, very high levels in the sampled sites from Antarctica too, in both the Antarctic Peninsula region, Mackay Glacier region, and McMurdo Dry Valleys region. And then if we compare the capabilities of these bacteria in the polar desert soils from those in agricultural land, forest land, grasses, and wetlands, we see that, consistent with our predictions, this capacity for using atmospheric hydrogen was continually enriched. Interestingly, carbon monoxide oxidation was similar to other regions. The capacity to use higher levels of hydrogen, as might be found through nitrogen fixation activity, was reduced. We also saw, interestingly, even though there's not much oxygenic photosynthesis in Antarctica, we did see evidence of this, these energy converting rhodopsins being highly enriched, indicating that this continual energy harvesting, both from atmospheric as well as solar energy sources, is important, even though water is limiting oxygenic photosynthesis per se. We saw very similar things in other hot and cold deserts, but then when we go to biocrusts and rocks, as you might expect, there was an even higher capacity for carbon fixation, especially through oxygenic photosynthesis, but interestingly, still a very high capacity for hydrogen oxidation and carbon monoxide oxidation. And so to take a bit more of a detailed view here, what was really clear from this global survey was that a really defining trait of desert communities, including in Antarctica, is this shift of the microbial community from heterotrophy, so using plant-derived vegetation, to autotrophy, so basically self-sufficiency by using hydrogen carbon monoxide to fix CO2. So for instance, we saw that we looked at 8 different carbohydrate active and peptide active enzymes. This is just showing 2 classes of them here, and we see consistently with hot and cold deserts as well as polar deserts a much lower capacity to use these carbohydrates and peptides, as you might expect, whereas we saw a much higher capacity to use hydrogen as well as fix carbon dioxide into biomass. Then we can also validate this with activity-based studies. We saw those much higher rates of hydrogen oxidation as well as CO2 fixation in desert soils. And in fact, it was the polar desert soils that overall had the highest rates of carbon fixation, although there were some exceptions to this rule, for instance, where we saw that, for instance, in the Atacama Desert, there was an incredibly high capacity for hydrogen-trophic carbon fixation, just as what we saw in the Antarctic ice-free regions, as I showed earlier in the talk. Another really interesting thing that came out from all of this analysis, again, focusing in on the Antarctic region, and in this case, we combined short read sequencing with long read sequencing, was that the capacity for trace gas oxidation has almost certainly been horizontally acquired in this continent and is subject to selection. So from beautiful work led by Laura Woods, we saw that, and there was evidence that the Nicoline hydrogenases, as well as other metabolic genes, had been horizontally acquired, including at the levels of both phylum and family. And when we do detailed phylogenomic analysis, these findings are substantiated. So I won't go into any detail here, and I don't think you can read this. But essentially, when we look at the hydrogenase and the rubisco in detail, we see a discordance between the gene trees and the genome trees, where we see this rapid dissemination of the hydrogenase and the rubisco across quite divergent taxa. And we also see, although it's not exactly definitive evidence, but we also see that some of these key metabolic genes, including the Nicoline hydrogenases, are in the vicinity of insertion sequences as well. One really interesting thing that's come up from this latest paper in PNAS is that while these bacteria capable of atmospheric chemosynthesis are the dominant community members in Antarctic soils, there are other things occurring too. So for instance, we saw quite high levels of Bordetella vibrio, we saw high levels of chlamydia, as well as Pitesibacteria, CPR, and Dependaceae. And these are all bacteria that are known to adopt, in the case of Bordetella vibrio, predatory lifestyles, in the case of others, symbiotic, probably parasitic lifestyles, indicating that in Antarctica, although the majority of life seems to be quite self-sufficient and is using these trace gases and other strategies to be able to survive despite the low energy inputs and all of that. We do see that at least some bacteria that adopt more predatory lifestyles or parasitic lifestyles can find a stable niche in the continent, indicating there's probably quite complex ecological food webs going on. And consistently, we also believe that the energy being derived from this atmospheric dependent carbon fixation is probably also feeding into higher organisms like springtails as well, but we have yet to prove that. So finally, I just wanted to take a little bit more of a view of some of the other regions we've looked at. So we've looked at whether trace gases are relevant for various other extreme environments, spanning everything from waters beneath the Ross Ice Shelf right the way through to the upper atmosphere and the lower atmosphere itself. And what we consistently see is that this capacity for trace gas oxidation is in various extreme environments has been selected for all sorts of reasons. And whether that be because light is unavailable, as is the case of below the Ross Ice Shelf, whether it's the case that there's extreme hydrodynamic disturbance, as is the case in certain sand ecosystems and whatnot. And to give a bit of a more detailed view of this, I wanted to just jump in briefly to our work in marine ecosystems, which in this case has been focused on the sub-Antarctic region as well as other regions, but it's probably relevant to the Antarctic region as well. And I think it will be incredibly interesting to do a more detailed study in the Ross Sea and compare across seasons to what extent these findings are relevant. So in collaboration with Sergio Morales and Fede Baltar, we did this survey of how do bacteria in oceans worldwide generate their energy. And this was led by Rachel Lappin. What we saw from this local transect, where we spanned 3 different regions, so tropical, temperate, and then this transect that expand from coastal to subantarctic waters off New Zealand, what we saw consistently was, although there was an incredibly high capacity for photosynthesis, as you would expect, these trace gases were quite important, with both carbon monoxide and hydrogen oxidation, the genes for these processes being quite enriched in certain sites. Then when we compared this globally with the Tara Oceans data set, what we saw was, again, this high capacity for trace gas oxidation throughout, alongside other processes such as photosynthesis. But interestingly, when you go into deeper waters, this is when you see this capacity for trace gas oxidation and carbon monoxide, hydrogen and carbon monoxide oxidation being really enriched. And we also saw that when we looked with latitude 2, we saw especially this capacity for hydrogen oxidation being both more abundant based on the metagenomes, more expressed based on the metatranscriptomes, but also more active based on activity-based studies. And so consistently, if we take waters back into the lab and we measure hydrogen and carbon monoxide consumption, what we see is that whereas carbon monoxide oxidation interestingly predominates in coastal and subtropical waters, perhaps indicating that in these regions where there's high levels of thermochemical and photochemical carbon monoxide oxidation, this carbon monoxide oxidation is serving primarily recycling role. In oligotrophic subantarctic waters, interestingly, hydrogen oxidation is most active, indicating that in these regions, hydrogen could be a significant source of energy. Now we need to really extend this to the Antarctic continent itself. But one thing that our collaborators, Fede and Sergio, have shown with Clara too, and this was an extensive collaboration with our lab as well, is that beneath the Ross Ice Shelf, there is actually a whole ecosystem that is primarily driven, again, by chemosynthesis. So what Fede and Sergio did here was 350 kilometers from Ross, from the Scott base. They drilled a gigantic hole, several hundred meters down, and then they took the metagenomes and metatranscriptomes of the bacteria present there. What they were able to show was that there was a very high capacity for nitrification, but also to a lesser extent carbon monoxide and to some extent hydrogen oxidation as well, indicating that even deep beneath the Ross Ice Shelf, which has very limited connectivity with the Ross Sea, this chemosynthesis in marine regions can sustain quite complex communities. And now what we're trying to do through the SAFE program, this is a large new program funded by the Australian government, led by Stephen Chown, is get a much better understanding of Antarctica's unique life and how this might change in response to forecast global and local change. And this involves 3 different themes, including a biodiversity theme, and which I'm primarily working in with insights from both climate change feeding in, but we're also using some of these insights to help support environmental stewardship in the region. So to wrap up, I hope I've convinced you that life doesn't need photosynthesis to prosper. Of course, this has been known for some time with this discovery that geological chemosynthesis can sustain very complex communities like these in hydrothermal vents. But in these ecosystems, at one time weren't even thought to be ecosystems, atmospheric trace gases can in fact power quite complex communities and provide an alternative basis for life. I think these findings have potential astrobiological ramifications as well. One of the mantras of astrobiology has always been to follow the water, but actually if we follow the gases, this might be a little bit more relevant given life can actually generate water from gases, but also life on Earth almost certainly originally evolved through chemosynthesis, not photosynthesis. So thanks so much for listening. I want to thank again my team, collaborators, mentors, and funding. Thanks so much.
Fantastic talk, Chris. Thanks so much. Hey, I just have one. Hey, no, I've got one question. In all of the work, or the majority of the work, was conducted in obviously hyper-arid or arid soils. What's your prediction for the for trace gas oxidation in general across a wetted gradient in an arid polar desert?
That's a very good question. We've looked at this to some extent in the Negev desert in Israel, where we went from subhumid north of the continent to the hyperarid south, and there we definitely saw that at least cell-specific rates of hydrogen and carbon monoxide oxidation greatly increased, as did the capacity. What we have also explored with this latest study was how do different deserts respond to hydration. And interestingly, in hot deserts, it seems that this atmospheric trace gas oxidation is stimulated by wetting, but interestingly, in polar deserts for regions we can't fully work out, and would love to have your insights into, we see that it's actually inhibitory, which is quite interesting. So I think there's a lot of work still to be done on those moisture energy sort of interactions.
Cool. We should talk about that. I think you're right. And another question that kind of is kind of connected to that is the horizontal transfer. Now, in an arid desert where you don't have connectivity, there's no liquid water cells to for transfer to take place. For the majority of transfer to take place, you have to have contact. If you don't have contact, how is that happening in a system that's not wetted and there's no connectivity between cells?
Yeah, that's a great question too. And I think one of the things with the horizontal gene transfer is we can't pinpoint exactly when this occurred. So even though there's evidence that there was this rapid dissemination, exactly when this occurred is unclear. But our central viewpoint is probably that that the main agents of turnover in these ecosystems are probably actually phages, and we believe that could possibly be providing some mechanism for horizontal gene transfer as well. But at this point is quite speculative. So we definitely need to explore that more. But I agree that probably in these regions, it's certainly the horizontal gene transfer that's happening is still, I say it's rapid, but that's rapid on probably scales of millions of years rather than the sort of time scales you might expect in, say, the open ocean and whatnot.
Mm-hmm.
Yeah, thank you so much, Chris. It's really very informative and it's very interesting talk. So I think I would like to ask Dr. Anil Kumar. I think he wants to give some suggestion regarding this talk and the further speakers presentation and all. Yes, sir, can you, would you like to speak something, Dr. Anil Kumar?
Yeah, I'm very happy to hear this talk, and it is very informative. And thank you so much, Chris Cary, for giving us a nice talk. So actually, we have to give some information about to the speakers about today's presentations. Time slot is given 12 minutes for presentation and 3 minutes for discussion. And all these talks will be recorded and it will be put in the past events in the SCAR website. And also it will be uploaded in the YouTube. So if anybody wants to, doesn't want to do it, then you can inform us through SCAR 2022@elbonmeetings.com. And the e-posters, it will be posted in the e-poster exhibition space. There, anybody can go there and make your comments. If you want to make any comments, you can do that. And any questions and answers, one session is there, Q&A. You can post your questions there, and that questions will be asked after each talk. Thank you.
Thank you, sir. So may I ask another 1 or 2 questions, because we are running short of time. So then we'll go to all speakers. And before speakers, I would like to ask the speakers, their presentations should be restricted with 13 minutes followed by 3 minutes discussion. Okay. So please have any 1 or 2 questions to Chris Grenny. Okay. Then
Tina. I think
okay,
Tina had a question. Yeah.
Yeah, hi. Great talk. Thanks a lot, Chris.
Thank you.
I was wondering if you think that this trace gas oxidation is just a survival mechanism, or can these cells actually grow and produce a lot of biomass based on that? I mean, I know that methane oxidation from the atmosphere has only recently been shown to actually generate biomass.
It is, Ali. That's an incredible question, and I think it's still an unresolved one. So traditionally, it was thought bacteria cannot grow from using trace gases alone, given the energy requirements their energy requirements would be too high, and the amount of energy generated from trace gas oxidation would be too low. I think this is true for the majority of bacteria, but I think there would be important exceptions. So bacteria with incredibly low energy requirements would be an example there, and keeping in mind that most of the energy requirements of cells are based on studies on cultured copriotrophs rather than the things that you might find in, say, Antarctica. But the other thing is, I think you sort of alluded to this, Tina, is when you have mixotrophy in the midst, then other things are possible. So there was that beautiful paper that I think you alluded to on atmospheric methane oxidation. And in this case, atmospheric methane oxidation is, they showed that these atmospheric methanotrophs are actually co-oxidizing hydrogen, carbon monoxide, and methane at the same time. Interestingly, the ones in Antarctica, though, they just seem to be doing methane oxidation only. But again, it's probably because the energy requirements there are lower given the lower temperatures as well associated with the continent. For the marine work, we've shown that hydrogen oxidation, even at atmospheric levels, can probably sustain chemolithoheterotrophic growth of a polar bacterium called Sphingopixis aliscensis. So I think, long answer, but in general, it will primarily be sustaining persistence, but for a subset of organisms, at the very least, mixotrophic growth.
Sorry, Chris, I am seeing one question from chat box. So the question is, any inventories for studying geological chemosynthesis in the Southern Ocean?
Can
I repeat?
Yeah, I don't fully understand the question.
Any inventories for studying geological chemosynthesis in the Southern Ocean?
I'm not quite sure what's meant by inventory, sorry, but I guess my view is for this initial study, we did use the Tara Oceans dataset, but there it doesn't really truly go into the Southern Ocean and the Ross Sea, and we would actually love to collaborate with anyone who does have access to either existing datasets or alternatively new datasets in the Ross Sea to understand, especially in those seasonal, variations, whether we see that enrichment of trace gas oxidizes as the lights go out in polar winter and whatnot. I also saw So I can't fully answer your question, Bibula, but I think it's a good question. And if you did have any insights there, get in touch through email, chris.greening@monash.edu. Sergey, that's a very good question. So lowest temperature limit for trace gas oxidation. With the McKay Glacier study, we went down to -20 and we saw very, very active processes there.
Thanks.
We'd love to try that at lower temperatures, but whether that works, yeah.
Yeah, thanks. Another question I'm seeing. So please quick answer. Lowest temperature limit for the trace gases oxidation.
Sorry, I just answered that one. So sorry, I'm probably doing your chair role. Sorry, but yeah, we don't know. At -20, it was very fast overall. Not super fast, but we saw quite strong trace gas oxidation. Yeah.
Okay. Thank you. So I have a couple of questions, but I will ask through later on through email or by some other way. So let us, Chris, thank you. Thank you a lot. And then now I can invite I would like to invite the next speaker, Christina Purkaria. And the title is Ice Microbiome Response to Thermal Stress. Yeah, speaker Christina Purkaria is there.
Dr. Mishra, I think she sent a video. I think
Video.
not in. Yeah. So can we play it now?
Yeah.
In the chat box, they have asked.
They have asked. Yeah.
Yeah.
I just did.
Yeah, I'm seeing. Yeah. Chat.
Hello, my name is Christina Porcara from Institute of Biology, Bucharest. My talk will highlight some aspects regarding the long-term and short-term impact of thermal stress on the ice microbiome. In the context of the rapid climate changes and knowing that 10% of Earth's continental surface is covered with ice, and an annual ice melting occurring for more than 5%, we are interested to understand what happens with the ice microbiome when shifting habitats from ice to soil due to glacier melting, and what is the effect of climate variation during ice formation on the entrapped communities. To answer these two questions, we investigated the structural and functional changes of the total and active ice microbiome in response to climate variations during ice formation, representing a long-term thermal stress, and submitted to a heat shock model experiment mimicking daily temperature exposure after glacier melting as a short-term thermal stress. This study was carried out based on DNA and RNA Illumina sequencing of 16S ribosomal RNA gene, ITS2, and RNA shotgun sequencing, meta-transcriptomic analysis, in addition to functional characterization of isolated bacterial strains. The habitat that was investigated was the perennial ice from Scarishvara ice cave. This limestone cave from Upper Jurassic is located in Carpathian Mountains in the northwestern part of Romania and harbors one of the largest and oldest underground glaciers identified so far that was carbon dated to more than 13,000 years before present. Sampling was carried out by vertical drilling from the top of the ice block and from the glacier surface, considering strata of different organic content, as we see here, organic rich and clear ice. The age of ice samples varied between recent ice of 1 year on top of the ice block to 13,000 years from the 25-meter ice core. For studying the climate impact on ice microbiome, we selected ice layers formed during war periods of 900 and 5,000-year-old ice and cold periods of 400 and 1,500 years old ice among the samples selected from the ice core and collected from the ice block surface, in accordance with the climate reconstitution data using stable isotopes. So these intervals were Little Ice Age, Medieval Warm Period, and Sahara Desert Formation period. The diversity profile of uncultured bacteria along the ice chronosequence indicated a variable phyla distribution, with actinobacteria dominating the Lea-formed ice and prevalent proteobacteria and bacteroidetes in ice deposited during warmer period, YP. In the case of active bacterial communities, both firmicutes and proteobacteria were highly represented in cold periods, while proteobacteria was clearly dominating the microbiome from the warm period ice layer. Different archaeal taxa were also majorly found in Lea and MWP formed ice, with preference for Crenarchaeota for the cold period and Euryarchaeota for warm period, with different functional roles, degradation of aromatic compounds, and metagenesis and ammonia oxidation for warmer periods, varying with the organic content of the ice strata. In the case of uncultured fungi, a clear split between Ascomycota dominating the warm period and Basidiomycota, mainly found in Lea-formed ice, was observed. In addition to the microbiome structure, the climate during ice formation seems to model also the rhizostome of the embedded bacterial community. In the case of bacterial isolates along the 13,000 years ice core, the antibiotic resistance profile showed a minimum during Lea deposition in blue, and maximum resistance during warm periods of 900 and 5,000 years old ice in orange, where the majority of strains showed a multiple drug resistance profile. To answer the second question of the thermal stress response of ice microbiome, the ice collected from a 900 years old strata from Cary was melted and submitted to 3 daily heat shock cycles, as we see, between 4 and 25 degrees, followed by 2 weeks incubation at 4 degrees. And the composition of active community and gene response during T3, T7, and T14 steps were compared with those of the untreated ice microbiome using RNA metatranscriptomics. Overall, we observed a major shift in the eukaryotes and archaeal community relative abundance after the heat shock cycles, which recover after 2 weeks incubation in the case of microeukaryotes. The structural response of active bacteria indicated a major shift after the heat shock in T3, in particular for actinobacteria, chlorobi, and firmicutes phyla, while proteobacteria and bacteroidetes were more resilient. For active fungi, the community appears to be more stable to thermal stress, except for Basidiomycota taxa that was more affected by temperature. The microeukaryotic community described here for the first time in a cave ice habitat appeared to be complex and was more affected by the heat shock treatment, T0, T3, in particular Chlorophyta taxa. Other phyla like Metazoa, Silophora, Cercazoa, Bicosoacida were more resilient. After post-treatment incubation in T7, Bicosoacida and Chrysophycea recovered during the first and second week of incubation, respectively. And this effect seems to being correlated with the decrease in bacterial community content appear to represent an active participation of protozoa in regulating bacterial abundance, limiting the growth of homeotrophic bacteria in organic-rich strata. At functional level, the heat shock appeared to also have a major impact on the active ice microbiome. The analysis of the differential gene expression here showed an overall transcript increase of the genes involved in translation, ribosomal structure, transcription, replication, and repair, most of them taking place during the heat shock steps T0, T3. Specific transcripts shift occur for genes involved in enzyme recovery, energy storage, carbon and nitrogen regulation, and cell motility, indicating a variety of molecular mechanisms of the ice microbiome for coping with thermal stress. Interestingly, the presence of GROL high transcripts in T0 showed a very fast response of the ice microbiome to temperature increase after the ice thawing and before exposure to the treatment up to 25 degrees, which indicates a short time for production of protecting chaperones. The carbon metabolites showed a differential gene expression of the carbohydrate binding, in particular glycosyl transferase and hydrolases, starting from T0, T3 step. Meanwhile, for the nitrogen metabolites, the heat shock step affected mainly the organic degradation and synthesis and denitrification processes, while the nitrogen fixation genes involving repair were upregulated after 14 days incubation for microbiome recovery. As a general wrap-up, these data reporting for the first time the presence of an active microbiome in perennial ice from caves showed a climate-driven profile of the ice in trapped prokaryotic and fungal communities regarding the biodiversity and antibiotic resistance of bacterial strains, and a complex structural and functional response to heat shock cycling treatment, giving a glimpse on the impact of glacier melting on cryospheric ecosystem in the context of climate change. I want to thank everyone involved in these studies, from the Institute of Biology in Romania, Emil Racoviță Institute of Speleology in Cluj-Napoca in Romania, the Swiss Federal Research Group in Zurich, Aarhus University in Denmark, and University of Antofagasta in Chile, and acknowledge the funding received from Romanian WFHC Agency and European Commission through the Eranet LAC project and Marie Curie ITN Micro-Arctic project. And thank you for your attention.
Yeah, thank you, Christina, for your video presentation. Okay, so if you have a question, we'll contact you through mail. So we can go the last for today's session. The speaker is Sofia Galavan. And the title is What Defines the Composition of Airborne Microbial Community? So I would like to invite Galavan, please.
Wait a second. Can you hear me and see the presentation?
Yes, yes, absolutely.
Okay. Okay. Hello, everyone, and thank you for joining us. I'm Sofia Galvan, and I'm doing my PhD in microbiology in Microbial Research Group from the Universidad Autónoma de Madrid, Spain. Our study focuses on the different ecosystems that can be found in the poles, especially Antarctica, and how they're connected by air. So during this talk, I'll provide you with some background on the microorganisms that we can find in the atmosphere and some of the results that we have been getting. So the atmosphere plays a crucial role in the microbial transport on the planet. The concentration of microorganisms that we can find per cubic meter of air are in the order of 10 to the 4 microorganisms over the land surface. Compared with other ecosystems, such as soils, for example, where values can reach orders of 10 to the 12 cells per grams of soil, these concentrations may seem very low. But despite this, given its size, the atmosphere is a habitat that houses a high number and diversity of microorganisms. The airborne particles that we can find in the atmosphere include from prokaryotic cells, small eukaryotes, fungal spores, pollen, to acellular structures such as viruses. However, the dispersion of microorganisms doesn't occur equally at all heights of the atmosphere. The flow of microorganisms is found mainly in the planetary boundary layer, the PBA. This atmospheric layer is characterized by containing a volume of air subject to a constant influence of the Earth's surface. This accentuated vertical mix allows the origin of microorganisms to be very diverse. Some processes that contribute microorganisms to the atmosphere are, for example, soil erosion, dissolution of drops of water, natural disturbances, how can be tornadoes, for example, or even anthropogenic disturbances, such as agriculture or sewage treatment plants. Once in the atmosphere, thanks to air masses, they can be transported for thousands of kilometers. And after this trip, finally, airborne microorganisms are deposited in new habitats through the gravitational effect, known as a dry deposition, or precipitations, rain, snow, known as wet deposition. It should be noted that the airborne community composition and its abundance are not constant in the atmosphere. Some studies have observed temporal variations, seasonal and daily, associated to the meteorological conditions and boundary layer height, and spatial variations depending on the sampling site or, for example, the path traveled by the sampled air masses. But why is so important understand the microbial atmospheric dispersion? This places us under the hypothesis made by Basbekin about the ubiquity of microorganisms. Everything is everywhere, but the environment selects. In this hypothesis, the atmosphere plays an important role because it's one of the fastest dispersal vehicles of microorganisms on the planet and indeed connects the most of Earth ecosystems. So the implications that the atmosphere has are relevant for many fields of study. For example, in epidemiology, as we sadly can see with the pandemic, plant pathology, with the pests that could spore entire crops, ecology, related, for example, with invasive species, or meteorology in relation to the formation of crystallization nuclei, among other fields. So despite all the important roles that the atmosphere has, this biome remains the less known on the planet. So let's go to the heart of this talk. But first, why we do our studies in Antarctica? Antarctica offers an ideal scenario for the aerobiology studies thanks to three mainly issues. In the first place, it's isolation with the rest of the world, although relative. In the second place, because extreme conditions make the development of life there a challenge. And lastly, is low degree of anthropic influence. In particular, this study focuses on the Antarctica Peninsula because in summer season is characterized by having a greater diversity than other Antarctic regions. It is due to all the possible biotopes that can be found there at summer season, I mean. A total of 6 samples were collected, 5 of them in Pearce Peninsula and the remainder in Avian Island. all the samples were collected as summer Antarctic campaigns to reduce the season variability in the urban community. In the case of air sampling, was carried out using a commercially available cyclonic collector, Coriolis, and then the microbial community composition of these samples was analyzed by 16S RDNA gene sequencing using amplicon sequence variants, ASVs. We use analysis based on ASVs because they make it possible to understand highly complex microbial communities in their entirety. I mean, this method captures all biological variations thanks to an error model that is capable of distinguishing between sequence readings with a resolution of just one nucleotide. And lastly, in addition to biological analysis, back trajectories of sample air masses were simulated with high split. So let's go to the results. To understand the samples at first glance, we look at the composition of the microbial community at the phylum level. So we can observe a total of 39 bacteria phyla with a dominance of these 5 phyla, similar to what was found in other aerobiology studies. The bacterial profile between the samples at this level is quite homogeneous, more or less. But if we delve deeper into taxonomy assignment, the situation changed. At a family level, the Bayer samples still maintain some homogeneity, but this time the avian sample different from the Bayer samples in its profile and seems to have more richness than the Bayer samples. At the same time, if we observe the alpha diversity index, we can also corroborate that. So at this point, we can find the first difference between locations. On the other hand, it should be noted that inside Bayer's homogeneity, we find a sample that presents greater richness and diversity compared to the rest of the Bayer samples, the B27. When we study the trajectories followed by the sample dermesis, we observe that all of them are characterized by a no-continental maritime route. When we refine further, we can see that the Bayer samples are grouped into 3 groups, 3 samples with a path through the Antarctic Ocean, the B25, the B26, and the B29, and 2 samples with Bellinghausen Sea back trajectories, one with a more oceanic passage, the B29, and another with a more coastal, the B27. In the case of the avian sample, it's characterized by air masses with a coastal Bellinghausen Sea route. So at this point, it seems to be a similarity between B27 and avian, which we think is, spoiler alert, is the great importance as we see next. Now, we will study in more detail the homogeneity between the Bayer samples that we observed and the possible similarities difference between avian and Bayer samples, but this time thanks to the ASVs. To study the homogeneity between Bayer samples, we apply the core 80 community, that is the ASV shared by almost 80% of the samples. The core 80 of Bayer samples is composed of 384 ASVs that represented more or less 60% of the total microbial reach in Bayers. So the homogeneity that we saw in the previous analysis about the urban community of Bayers is confirmed. But what is it due to? As we have seen, different studies show that there are temporal and spatial variations in the urban community of a place, right? So our first hypothesis that will explain this homogeneity of cells between the Bayer samples and the avian sample doesn't have, as we will see, is the spatial influence by local biotopes. Remember, all the samples were collected at the same season. So therefore, we compare the Bayer's core 80 with a pool of Bayer samples from one of the most abundant local biotopes at these seasons, the free ice soil. The results, almost 8% of the ASVs that make up the core 80 have been recorded in the soil pools, confirming our hypothesis. The biotopes surrounding the sampling site exert a local influence to the urban community and therefore confers difference between communities of different locations, as we can see here. If we compare avian versus Bayer samples, including the soils, using beta diversity analysis, we can see that in the first dimension, the samples are grouped by locality. I mean, there is an influence of the sampling place. However, we can find similarities between the locations too at the level of ASVs. About 80% of Bayer's core AT ASVs have been observed in avian. So it seems that there is an influence shared by both locations. Our hypothesis, it could be due to a regional influence, Antarctica Peninsula by the Antarctic Peninsula, or continental influence. However, if we go deeper in this comparative and we observe the percentage share between avian and its Bayer samples, around 2% of the ASVs of its samples are shared with avian, except one sample, the B27, with a higher similarity to avian than the rest of the Bayer samples. This exceptionality of the B27 sample stands out even more when the exclusive ASVs of its sample and their presence in the avian sample are studied. According to the results, and thus observed in the back trajectories, everything indicates that the airborne community of both sample is influenced by the coastal travel that sampled AMSs did. That is, the exclusive microorganisms, I mean, the exclusive ASVs of both samples could be due to a coastal regional source. In short, our first results are a contribution to understanding the composition of the airborne bacterial community of Bayer's Peninsula at summertime, and their possible geographical influence, local, as we saw in the core AT and the comparison with the soil, and regional and continental influence, as we saw with the comparisons between locations. However, microorganisms dispersions is still very unknown, and we believe it is necessary to continue with monitoring it around Antarctic continent, as well as in the rest of the planet, with a multidisciplinary perspective that include biology, physics, methodology, and statistics. So thanks for your attention, and I will be happy to answer any question.
Thank you, Sophia. It's a really nice presentation regarding the airborne microbial community. So I would expect a couple of questions. Yeah, please, Greg, Professor.
Sophia, that was a wonderful presentation and a beautiful piece of work. Hey, I just have one small question, and that in looking at your you've talked about a lot about abundance, but looking at the minor components, and this is something that's close to my heart, did you pick up any thermophiles, that at least known thermophiles, in any of the minor components of your surveys?
Can you convert the question, because I don't understand?
So did you pick up any ASVs that might be associated with high temperature environments?
Oh yeah, indeed, I am working on it, and I compare all the core AT of Bayer's with the studies, but not only with the Antarctic studies, even with the Spanish studies, or for example with the Arctic too, and different ecosystems, to have more expanded view about what is happening.
So we found in the dry valleys, which is obviously a long way away from where you are, we routinely pick up in the background. So these aren't core members; these are just in minor numbers in the back. Okay. These are minor reads, maybe on the orders of 10 reads in a given library. We pick up high temperature bacteria, and we're seeing this pattern along the entire Victoria Land coastline. But I'm curious. Well, you've got Deception Island up in the peninsula, well, up in the sub-Antarctic. Is probably the most local geothermal source, but it's something to keep an eye out for. And if you find any, let me know. Okay.
Okay. I search with you.
Thank you. I'm seeing that Dr. Fed is here, Federico Baltar. So Can I ask Fed to give any comments?
Yeah.
Fed.
Hey, if I could interject very quickly, Sophia, awesome that you use the Coriolis in the Antarctic Peninsula. In the dry valleys, I actually collected with the Coriolis as well, so Steve Archer. It would be really interesting to actually compare those airborne samples that we collected in the valleys with your peninsula samples to see whether or not there's, one of your big questions was the continental influence, and I think that would be a really interesting way of just seeing if there is any sort of overlap going on there.
Yeah.
So I'll send you an email in a bit, but it would be really great to continue. Thanks very much for the talk.
Totally. I totally agree.
Thanks. Dr. Baltar. Hello, Dr. Baltar.
Yeah, hello. Hi, Mishra.
Yeah, I'm seeing you so for a long time. Okay.
Yeah.
So, do you want to give any comments or questions, something else?
No, I think I'm good from my side. Yeah.
Okay. So then I would like to request all, please switch on their videos. So we'll have one snuff. We'll take a snuff. I would like to request to all. I would like to request all. Yeah, Dr. Sabu, can you please take a snapshot?
Done.
Done?
Done. Thank you.
Okay, thank you. So today we are now finishing this first slot of the session 26, ecological implications and adaptations of microorganisms in the polar environments. So we finished 5 talks, 5 presentations, including the keynote talk. So tomorrow again we'll continue the same session, and we'll have another 1, 2, 3, 4, 5, 6, 7, another 8 presentations are there. So I would like to request to all to be attend at 5 to 7 UTC. 5 to 7 UTC. Okay. Anything else? Anybody? Any comments?
Everything fine, Mishra? Everything went
Okay.
well. Thank you so much for conducting this session.
Okay. So we'll meet again by tomorrow. I mean 5 to 7 UTC.
Yeah.
Okay.
Thank you.
Thank you.
Thank you all. Thank you, Walter. Thank you, Professor Craig, and to other speakers, all speakers. Okay. Thank you so much.
Thank you.